Hi, everybody. I'd like to thank you for joining in. My name is Alana Jacobson, and I'm a research entomologist at Auburn University. Today, I'm going to be providing an update on what we've learned about aphid vectors, virus incidence, and management of an emerging aphid-transmitted cotton virus, cotton leaf roll dwarf virus. I'd like to start off just by acknowledging a lot of hard work and aphid counting done by my lab group, and funding sources, including Auburn University, industry funding sources, and also government-funded programs. So today I'm going to be providing information about an emerging aphid-transmitted virus that was first detected in 2017 in samples collected in Alabama and is now reported across the cotton belt from Virginia to West Texas. This has been a tricky problem to understand and study because there's a lot of variation in symptom expression. Some of the common symptoms that you'll hear about are shown in pictures at the bottom of this slide. And plant pathologists, after looking at this problem for 2 years in the field, are finally of a consensus that you cannot identify this virus based on visual symptoms alone. So if you want to know that you have a CLRDV infected plant, laboratory testing is required. So there's been a lot of variation in virus symptoms. There are also asymptomatic infections that occur, and there's also a high degree of variation in yield loss. We've had some cases of extreme yield loss where in September there were not any bolls on the plants, but this is only a handful of fields that we've observed. And by far and by large, most of what we're hearing from people is that you either have symptomatic plants or asymptomatic plants, but no apparent impact on yield. So from a research perspective and trying to assess what type of risk this poses to the cotton industry in the United States, we're basically approaching it from two different perspectives. One is we're trying to conduct research to understand what's the risk of plants becoming infected with cotton leaf roll dwarf virus. And the second part is we're trying to identify factors that contribute to disease and yield loss outcomes. For the first part of my talk, I'm going to focus on this first bullet point. So I'm going to start off just by providing some background about what we know about how insect-transmitted viruses are spread in the landscape and what we know about these different factors with respect to cotton leaf roll dwarf virus. And so to start out, basically spread of a virus into your crop is going to be determined by several different factors. And with respect to landscape-related factors and vector-related factors, how many species of aphids that are transmitting this virus is going to be important. How well and how quickly they transmit the virus is important. How large are the population sizes of these aphid vector species and what proportion of them are actually transmitting the virus in the landscape is important. And then we also need to know about where they're acquiring the virus from. So what are the alternate hosts and what are the seasonal population dynamics of the aphids acquiring the virus and then transmitting it to the crop. There are also other factors that are important, such as distance of the crop from these alternate sources, and typically the greater the distance, the lower the virus spread. So moving on to what we know about these factors with respect to CLRDV is we're in the process of trying to get a better understanding of what the full host range of CLRDV is, but some initial testing has confirmed cotton leaf roll dwarf virus in this list of hosts. And as you can see, these are very common weed hosts in the landscape. Now moving on to what we know about aphids in cotton. Here is a slide that shows all of the species of aphids that are reported to infest cotton in the United States. Now, cotton aphid is the species we talk about most because this is the one that will colonize and build to high numbers in cotton, where the other species do not seem to persist for very long. Cotton aphid is also the only reported vector of cotton leaf roll dwarf virus in the Americas. However, just because these other species don't persist in cotton, from a virus transmission perspective, doesn't mean they can't contribute to spread of the virus from those reservoir hosts to cotton. And that's because an aphid only has to feed on a plant for long enough to transmit the virus. It doesn't actually have to colonize and persist on the plant. And what we know about transmission of cotton leaf roll dwarf virus by cotton aphids is that it can occur very quickly. There was a study in Brazil that characterized the amount of time it took for aphids to transmit the virus. And they found that alates, which are these winged forms of aphids, which you can see in this picture right here. So these alates that have wings are responsible for dispersing throughout the landscape and colonizing new hosts. And this morph of the cotton aphid was reported to transmit in under 40 seconds. And I have a graduate student that confirmed this result using Alabama populations of the virus and the vector. The other thing that you need to keep in mind when thinking about virus spread in the landscape is that this is what we call a persistent virus because it actually is retained in the vector body for long periods of time. And what that means is that the vector does not shed the virus very quickly. In Brazil, they observed that aphids could transmit the virus for up to 12 days after acquiring it. My student observed that they could transmit for up to 23 days after they can acquire it. So essentially, virus transmission can occur over the lifetime of the aphid. The other thing to note about these cotton aphids and other species of aphids that infest cotton is that they have very broad host ranges. And basically what this means is that there's going to be a greater chance that they will have a host that can become infected with the virus. So to better understand the risk of virus infection to cotton, we need to understand the number of weed hosts, we need to understand the number of vector species, and we need to understand the seasonal dynamics that basically define when those vector species move from these reservoir hosts, disperse through the landscape, and infest and subsequently infect the cotton crop. And so this initial movement from the weed host to the crop is what we call primary spread. Once we have virus transmission and aphid establishment in the crop field, we then need to understand, as those aphid populations are building and spreading through the field, are they also spreading the virus through the field after colonization? And this within field virus spread is what we refer to as secondary spread. Other factors that are important in this equation is the timing and how that relates to crop age, because generally in different pathosystems in the southeastern United States, we see that younger crops are more susceptible to virus infection, whereas older plants have some level of mature plant resistance. We also need to understand the magnitude, so the numbers of alternate hosts, the numbers, the population size of the aphids, and basically the larger those two things, the higher the risk of infection. Another thing I wanted to mention is that there can be variation among different geographical areas, and that's because a lot of these different factors related to the reservoir host and the aphid are also governed by environment, and we have very different environmental conditions across the cotton belt. Even here within Alabama, we see a lot of variation in cotton leaf roll dwarf virus incidence across the state, where we see very high incidence in the southern part of the state, and as you move north, you generally see a decrease in incidence. And so it's important to understand these factors so that we can understand this variation and define areas that are at risk for this problem. And we also need to understand how much virus spread is actually occurring in the landscape and when it occurs, because any type of virus spread is important and could potentially be influencing risk. So this could be virus spread that occurs in the weed hosts. This can be virus spread that occurs within the crop to increase numbers. The cotton crop might also serve as a source for virus that moves back out into the landscape. And as an example of this, I wanted to show a slide that shows a field late season after harvest. Basically, cotton stalks in this field all had regrowth in them. And this was a field I came across in central Alabama in late 2019. Every plant had regrowth, every plant had aphids. And just kind of as a mental exercise, if you go with, there were approximately 30,000 plants per acre in this field, about 5 aphids per plant, which is probably an underestimate, but we'll just go with that. And it was probably about a 150-acre field. This 150-acre field could serve as a source of 22.5 million aphids in November that could then disperse throughout the landscape and spread the virus. And in central Alabama, we typically see incidences of 50 to 70%. So there could be a significant amount of spread occurring at multiple times of the year. So now I wanted to move along and talk about some of the research that we've been conducting and what some of the results of this research is. We've been conducting trials over the last 2 years, trying to better understand these aphid vectors, the timing of virus spread in the landscape, and relating that to the aphid species that are present when it's occurring, and also trying to conduct some trials that try and identify other factors that increase the risk of infection to the crop. So to monitor for aphids, we've been setting out traps. These are just little yellow pan traps that have a solution of propylene glycol in them. The aphids are attracted to the yellow, and so they fall into the solution and we can collect them. And we have these traps out in the field for a week around cotton fields at experiment stations at different locations. To monitor virus spread, we're using 2 to 3 true leaf cotton. And so basically this is the idea that young plants will be susceptible and that any aphids flying through the landscape could potentially be landing on these plants and transmitting the virus if they're carrying it. So they're basically indicators that are placed out the same weeks as our aphid traps. They're collected, then taken to the greenhouse and grown for a couple of months until we can test them for virus infection. I'm going to start off just by showing you some of the data that we've gotten from these trap runs. We're learning a lot about species composition, and so in this slide there are pie charts that show you the relative proportion of the total number of these different species that we have collected in Alabama and Georgia in 2019. Here's the list of the aphids that infest cotton in the United States. We did not catch one of those species. And you can see that the majority of species at both locations is indeed the cotton aphid. Other pieces of information we're getting are about the seasonal dynamics of these species. And so all of these graphs looking at aphid trapping are going to show the same, have the same layout, and that'll be the total number or the average number of aphids per trap on the y-axis and the dates that we trapped on the x-axis. And what you can see is the total number of aphids through time that are captured. We see this peak of aphids, this dispersal event that occurs late June or early July in Alabama. I'm not going to show the data from Georgia for the sake of time, but it has a very similar pattern. This gray area is the proportion of those total that were cotton aphid. And you can see that during this peak dispersal event, cotton aphid is the predominant species. The other thing I have on this graph are these little asterisks, and this shows you the weeks that we were able to detect virus infection in our sentinel plants. So these are weeks that virus spread was detected in the field, and this coincides nicely with cotton aphid dispersal. The other thing I wanted to show is that early in the season, so weeks 1 and 2 after plant, we also saw virus spread occurring in the landscape. And interestingly, captures of cotton aphid were very low at this time, and instead we had high numbers of this corn root aphid. We do not know if the corn root aphid was responsible for transmitting the virus, but this suggests that we have another vector in the landscape. Here we also detected virus transmission occurring late in the season, and we're not quite sure what species was responsible for this spread, because captures of all species were low at this time. This could potentially indicate secondary spread that was occurring from our cotton plants into, so from the cotton field we were trapping around onto the sentinel plants that we were using to monitor virus spread. In 2020, we expanded the area and the number of locations for aphid trapping with the help of cooperators from across the cotton belt and states where CLRDV has been detected. And on the next slides, I'm just going to show you some general trends. So we don't have any of the species identified yet, but what we do have are the total numbers counted. And what I'll show you are two graphs that just kind of show you some of the general trends and seasonal patterns. And the first thing I'd like to point out is that there is a lot of variation among these different locations. On these two slides, I tried to group them by locations that showed similar type patterns. And so on this slide, it looks like these locations, there's a little bit of activity early in the season. So similar to what we saw 2019 in Bruton, where we had that early flight of predominantly corn root aphid. So it'll be interesting to see what species are here. We also saw this June-July flight timing for aphids, and a little bit of seasonal activity late in the season, but not a whole lot. I also wanted to mention that the other locations outside of Alabama where they were able to actually monitor for CLRDV spread using sentinel plants was College Station, Texas, which is the brown line. And I just wanted to point out that in College Station, they did not detect any positive tests for CLRDV all season long. And that coincides nice with the fact that they don't see a lot of CLRDV, or they didn't in other field trials they conducted. In contrast, I don't have the data up here, but in Alabama, we did see similar patterns of virus spread as we did last year that occur both early in the season, April and May, also June and July through August and September. The next locations I'll show you are on this graph, and I have Tennessee and Mississippi up here just because I couldn't put them down here without them being hidden. But at both of these locations, interestingly, we had very low captures of aphids all season long. We also had a low number of aphids in South Carolina, whereas in North Carolina, North Alabama, we saw larger populations of aphids. And again, there appears to be some sort of early season and also late season flights of aphids, but the timing is variable across all of these locations. So right now we're in the process of identifying these aphids, and we're continuing trapping and sentinel plant monitoring in 2021, trying to get a couple years data to try and figure out what some of these patterns of virus spread and aphid dynamics are. The last research I wanted to talk about before I conclude my talk are some research that we've actually been doing to try and identify what factors are contributing to disease and yield loss outcomes. So in Alabama, over the last 2 years, we've been able to conduct a cage study. And basically what this is it's 2-row plots that are about 20 feet long that we put cages over so that we can actually manipulate the timing of an infection by introducing virulliferous aphids, so aphids that are carrying the virus, into these cages. And by doing this, we're able to exclude the virus, which we can't currently do in the landscape because we get 50 to 100% infection in small plot trials that we conduct. So by caging, we can have these different treatments where we can actually look at disease severity and yield in plots that did not have aphids, had aphids that did not have the virus, and we can also release them over different dates so that we can look at interactions between plant age and the timing infection on the severity of either disease or yield loss. And so in 2019, we released them a week after emergence, 2 weeks after emergence, and 3 weeks after emergence. And so what this looks like, the plants grow very nice in these cages. It's a little bit warmer than it is in plots where you do not have the cage, but that basically corresponds with 3 to 4 true leaf stage, 5 to 7, and first pinhead square in these cages. And you can see that we just introduce a plant that's known to be infected, that's infested with aphids, and we spread them around the plot. And we actually ended up with good levels of infestation and good levels of virus spread in these different plots. After the aphids transmit the virus, we then eliminate them from the crop by spraying insecticides, and eventually we have to remove the cages because the plants get too big and outgrow them. There's a lot of data that we collected from these. We have aphid counts, we have plant mapping data, but what I mainly wanted to focus on was data we collected on yield. So this is our yield data from 2019, and what I basically want to point out to you is there was around a 400-pound difference that was statistically significant between the plots where we released aphids a week after emergence versus plots that either were infested with aphids that did not have the virus or were not infested with aphids at all. And it was interesting because the yield loss was not due to a reduction in the number of bolls. As I said, we had plant mapping data that went along with this, and if you look at the total number of first and second position bolls on our final plant map date, they were not significantly different among treatments. The other thing is there seems to be some sort of a numerical trend where as crop age increased, we had an increase in yield as well. So the earlier the plants were infested, the more severe the reduction in yield. Interestingly, when we repeated this trial in 2020, and in 2020 we did not see the same reduction in yield that we saw the preceding year. And the other thing I wanted to mention about these trials is we didn't really see any apparent symptoms of disease either. And so I think the variation in data that we've seen across the 2 years, along with the lack of clear, obvious symptoms of cotton leaf roll dwarf virus disease, just basically speaks to the fact that we need to do some research and better understand what types of interactions are occurring between these cotton varieties, virus infection, and the environment that actually lead to symptom expression and yield loss. So to put it all together, we have confirmed in lab trials that the cotton aphid is the vector of CLRDV in the US, and we're generating a lot of good data about seasonal dynamics and virus spread that are helping us to identify other aphids that we need to test to see whether or not they can also transmit the virus. We're also seeing a lot of variation in those seasonal dynamics and incidents, and these seasonal patterns of movement and differences in both the number of species moving and potentially species composition might be what's underlying all of these differences in incidents we're seeing across Alabama and the rest of the Cotton Belt. And so we hope to continue these trapping efforts again, at the very least in 2021, and get a better handle on what these drivers of virus spread are and how variable they are across the Cotton Belt. Once we understand those, we might be able to devise some agronomic practices or implement some agronomic practices that might reduce virus spread. We're also continuing research studies like the cage study and coming up with other field trials that we can conduct to better understand what types of environmental factors or agronomic factors might be contributing to both virus incidence and potentially contributing to symptom severity and yield loss outcomes. So with that, I'd like to thank you for your attention and for listening to this talk. We've got a great group of collaborators that are working across land-grant universities in all these states on the entomology, plant pathology, and agronomy side of things, generating a lot of data, and hopefully we'll have good updates again for you next year. Thank you.
Greetings from the UGA Tifton campus. This is Phillip Roberts, Extension Entomologist with the University of Georgia, and today I would like to continue our discussion regarding management of cotton aphid and implications for cotton leaf roll dwarf virus. Cotton aphids are distributed across the world and vary in size and color from a light yellow to a dark green or almost black. These highly prolificous insects, meaning they have many host plants, will typically infest the undersides of leaves. They have parthenogenic reproduction, meaning that females do not mate and will actually give birth to living young. They also have a very high reproductive capacity. One female can give birth to over 80 offspring, and a newborn aphid will begin giving birth to aphids in 4 to 5 days. And for this reason, parthenogenic reproduction, high reproductive capacity, this pest is very prone to develop insecticide resistance. Now, we also have winged form and wingless forms. The formation of wings is typically in response to crowding or poor host quality. Now, cotton aphids can be found on cotton at any time during the year. Here's a picture from 2019 on cotyledon cotton. This is literally 4 days after planting, and you can already see we've had some winged aphids moving into this field as cotton was emerging. This is just a little later in time, one leaf cotton, but again, you can see where winged aphids have infested the field and are giving birth to living young. Now, we had never paid a lot of attention to aphids on seedling cotton prior to cotton leaf roll dwarf virus, but the more we looked, we realized it wasn't uncommon that we could have these infestations very early in the season. Now, we're most accustomed to seeing aphids on squaring cotton and flowering cotton. These numbers can build to, populations can build to very high numbers. You can see in several of these pictures, the leaves appear shiny. That is just honeydew, which is a sugary excrement excreted by the aphids. On that honeydew, sometimes we may observe a sooty mold, which can develop on those sugars. If we have severe infestations for an extended time, we can see yellowing in the terminals and some stunning of plant growth. Now, typically in the southeast, and especially in Georgia where I'm located, we will see a naturally occurring fungus, Neozygotes freseniae, which will come in and cause the aphid populations to crash. Now, we associate this naturally occurring fungus with these gray fuzzy aphid cadavers on the bottom sides of leaves. And once we see this fungus in the field, we usually expect populations to crash within about a week. Now, after the aphid populations crash, they can rebound to detectable levels later in the year. On the left, you see a picture that was actually taken on August 20th. The aphids had crashed early in July. But again, the more we've been observing cotton over the last couple of years, we've recognized that it's not uncommon to see low levels of aphids in fields about every field we visit. Now, if these aphid populations start to become established, typically the fungal episodic will come back through and cause the populations to crash. Even after the season, even when we have destroyed stalks or mowed stalks in the extreme southeast, we can see regrowth on these cut stalks, basal regrowth during the winter months, late fall months. We'll actually see aphids on these regrowth in fields. Aphids really don't overwinter, they just keep moving from host to host during the winter months. Now, there are many commonly occurring weeds that we know are hosts for aphids. I've listed several here. Some of the more common ones are highlighted, such as Carolina geranium, henbit, and primrose. But this is some data that was collected back in the 40s in Louisiana. But the point I want to make is that cotton aphids utilize a multitude of hosts, cotton being one of those. There are many other crops we produce, vegetable crops, that are cotton aphid utilizes, as well as some of these commonly occurring weeds. So as we've discussed, cotton can infest cotton from emergence up until the crop is defoliated. And in Georgia, cotton aphids typically infest 100% of our cotton at some level. Now, even though they're a very consistent pest, in some years they will build to high numbers, and it's obvious that we do need to treat some fields for aphids each and every year. But research in Georgia has not demonstrated a consistent response to controlling aphids. Now, we have some very effective insecticides in terms of lowering plant stress, but most of our growers, and our normal recommendation is to wait on the naturally occurring fungus, which will cause these populations to crash. Now, back in the late 90s and through 2008, over a period of about 10 years, we actually did a lot of work looking at the impact of cotton aphid on yield. And actually, this is a mean yields from 27 different trials. And basically, a lot of these were on-farm strip trials, but we compared an untreated versus treated. Now, these treated fields range from 1 to 4 sprays, but the bottom line is we did not see a significant difference in yield from treating aphids. There was only a 5-pound difference over those 27 trials. But again, there are going to be specific situations where cotton aphids will likely need to be controlled. So our management strategy has always been to conserve beneficial insects and hopefully wait on the naturally occurring fungus. But again, we have some very effective insecticides in terms of reducing plant stress from cotton aphid. So in recent years, since the detection of cotton leaf roll dwarf virus, entomologists across the cotton belt have actually are revisiting aphids, attempting to learn more about their biology and ecology, and whether management of aphids could impact the incidence or severity of this virus. A group of southeastern entomologists, we refer to ourselves as a Southeast Row Crop Entomology Working Group. Over the past 2 years, we've done what we call a regional project at these various locations across the Southeast, just looking at a multitude of insecticides to see how or which products are most efficacious. This was a regional project funded by Cotton Incorporated. Basically, these were standard small plot replicated trials. We went in and sampled the 4th main stem leaf below the terminal, and we had hoped to count aphids at 4, 7, and 14 days after treatment. Now, what we've done is I've calculated a percent control for all of these locations. Yield was taken at some locations, but that was an optional data. Now, what I've done here is taken the mean yield for each of these treatments from any trial which they occurred across this Southeast Regional project. Now, we can't really make direct comparisons because some treatments, for example, Brigade was only in 7 locations or 7 trials, whereas some of our other treatments, such as Transform, was in 12 trials. So what I've done is just calculated a mean percent control and put a standard error bar for each of those means. And what you see, we do have several insecticides here at 3 to 5 days after treatment, giving us anywhere from 70 to 90% control. Now at 7 to 9 days after treatment, we start to see a kind of a general break amongst our products. Some of our more recently introduced products tend to perform better than some products that have been around for several years, such as imidacloprid, for example. But again, We're just looking for trends in the data, but it looks like the products we're currently recommending in a lot of states are some of our better performers. At 14 days after treatment, we're still seeing some level of control, but again, our number of trials where we're able to get data at 14 days after treatment is much less. And in most situations, that was just due to that naturally occurring fungus coming in and crashing those populations. Now, we can make statistical comparisons of a few of our treatments. So what I've done here is basically pulled out treatments that are currently recommended in Southeast cotton producing states. And if you see here, products such as Transform and Sail and Carbine and even Bidrin provided statistically greater control compared with Admire Pro and Centric. Now, Bidrin tends to be erratic when used on the farm for aphid control, but in these trials it performed pretty well. At 7 to 9 days after treatment, again, we see those same 4 products providing significantly greater control than Admire Pro or Centric. At 14 days after treatment, again, the same story. So I think products which we're currently recommending do tend to give us our best control. And I would say even the products that didn't give us the greatest control, such as Admire Pro and Centric, in many situations they still relieve some of the stress put on the plant by aphids. Now, one thing I will say is when we were compiling this data, there was a lot of variability in how these insecticides performed. And I really think there are 2 reasons. Number one is just insecticide susceptibility. That's going to vary by state to state and even vary within regions in the state. And the second thing is just reinfestation after an application. When was the trial initiated, or when did you make the decision to treat aphids? That can influence performance of these products. For example, this is some data from Dr. Jacobson, but basically she has surveyed many populations of aphids in the state of Alabama and looking at their susceptibility to imidacloprid. 95% of those populations had a resistant ratio greater than 10-fold. 50% of those populations had a resistance ratio greater than 100-fold. Timing of when you treat. Dr. Jacobson showed you this data earlier from 2019, but this shows the number of cotton aphids collected in a pan trap. And what you can see if you focus on this red line was in Tifton, Georgia. We have a lot of aphid movement during the last week of June and the first week of July. Here's a picture of a field which we had treated with a very good aphicide on June 21st and June 27th. I took this picture 4 days after treatment, and you can see we just had a massive migration of adult or winged aphids coming into this field, and they immediately started giving birth to offspring. Even at 4 days after treatment, in this situation where we had this massive migration occurring, we couldn't really see that we gained any control of aphids. So during 2019, we conducted field trials in Alabama and Georgia to investigate whether aphid management reduced final incidence of cotton leaf roll dwarf virus. We did this by manipulating aphid populations in one of 4 ways. Number 1, we had an untreated. Number 2, we tried to eliminate aphids. We began treating these plots with acetamiprid, which is a very active insecticide for aphids. We began applications at the first true leaf and sprayed weekly throughout July. The next treatment we sprayed when we first observed aphids in a field. And again, then we sprayed it weekly. And the third treatment, or fourth treatment I should say, is we just made a single application around the 1st of July. We did these aphid management treatments on 2 planting dates, a May and a June planting date. Now this is a busy slide, but what I'm representing is the percent infested plants. And this was in Bruton, Alabama. This was Dr. Jacobson's trial. But what I want to point out, whether treatments were being sprayed weekly or not, 100% of the plants had at least some aphids on them for a 2-week period. We saw a similar result in Georgia. This graph is set up a little differently. The solid lines are the untreated. The dashed lines are where we were spraying weekly, beginning at the first leaf. The blue is our May planting date. The orange bars are our June planting date. But the point I want to make, whether we were spraying weekly or not spraying at all, there's very little difference in the percentage of infested plants. So yes, we can reduce the number of aphids, but we can't eliminate aphids, at least by spraying weekly. Now, in the latter part of July, we did sample plants from these trials, and we tested them for the presence of cotton leaf roll dwarf virus. So this is the mean percent of plants that were confirmed positive for cotton leaf roll dwarf virus. So in Bruton, Alabama, in the untreated, 80% of the plants tested positive for the virus. If you compare that to where weekly sprays were initiated, 84%. There's really no difference in the incidence of virus. The same could be said for the trial conducted in Tifton, Georgia. 99% of the plants were confirmed positive for CLRDV. In untreated plots, 95% of the plants tested positive for the virus where we were treating weekly. There was also no significant difference in yield on either planting date based on use of insecticide in Alabama, and the same could be said for trials conducted in Georgia. Now during 2020, we did conduct a trial similar to that was conducted in 2019. We actually had 3 planting dates. We planted in April, May, and June. We had an untreated, and we had plots where we treated weekly again with acetamiprid. What I'm showing in the left-hand graph is the percentage of plants confirmed positive for cotton leaf roll dwarf virus in the April plantings, untreated in the blue, weekly sprays in the orange. So April, May, and June planting dates. There's basically no significant difference whether we treated or did not treat for aphids in terms of the percent of plants that tested positive for the virus. Likewise, there were no significant differences in yield for any of the planting dates where we treated aphids compared to where we did nothing for aphid control. So that gets us to a conundrum. We had a lot of plants in these trials that were testing positive for cotton leaf roll dwarf virus, but we had very few plants in these trials where we actually saw symptoms or disease. So there's a big difference whether the plant tests positive for the virus or whether it is expressing symptoms and has cotton leaf roll dwarf disease. And again, in these trials, we had very, very few plants that showed any symptoms or yield loss associated with what we believe to be cotton leaf roll dwarf virus. Just want to continue building on some trials we've conducted in Georgia. This is a trial, I call it a pheno trial. I'm actually initiating trials or treatments at different times during the year and aggressively protected. The way we set these up, there's 6 row plots by 30 feet in length, randomized complete block. It ranges from an untreated plot to plots which were treated 7 weeks, 7 consecutive weeks, initiated about the first leaf on our most aggressively protected plot. We'll go in and count the number of aphids prior to treatment so we know what was there. But again, we're just trying to initiate aphid control at different times during the year. So most years we will see the naturally occurring fungus come in and cause our populations to crash sometime around the first or second week of July. So what I'm illustrating here is actual aphid counts in all of these plots on a specific date. In 2019, we made these counts on July 2nd. So just to set this up a little bit, if we look at this blue bar on the bottom left, this cotton was planted on May 14th. Well, on the 30th of May, I began treating this cotton once a week. This cotton had actually been sprayed for 5 consecutive weeks when we made these aphid counts. If we look at the second bar, we initiated our first spray on June the 6th. So it had been sprayed 4 consecutive weeks, 3 consecutive weeks, 2 consecutive weeks. In this bar, we initiated treatment on June 27th, so it had only been treated 1 time. The same can be said for 2020. The orange bars are untreated on the date at when we made this aphid count. But the point I want to make is we were unable to eliminate aphids, even where we sprayed weekly for multiple weeks in a row. So if we're unable to eliminate aphids, we're unable to eliminate the risk of transmission of this virus. But I think that's an important point that we just understand. We can achieve very good control, 90+% control of aphids, but we cannot eliminate aphids with insecticides. Now, over the course of 2019 and 20, across the Southeast, most of these trials were in Georgia, but I have summarized yield data of untreated plots versus treated plots. Now, for the treated plots, if these trials were part of the regional trial with all the different insecticides, I just took a mean of all of the insecticides to compare with the untreated. But the bottom line is in 2019 and 20, the response from controlling aphids in terms of yield is very similar to what we saw 10 to 20 years ago. Again, no statistical difference in yield. Actually, it was 5 pounds difference 20 years ago. What is that? 7 pounds, I guess, the last 2 years. So in the absence of virus or cotton leaf roll dwarf disease, it doesn't appear that control of aphids is going to help us in terms of yield. So one thing that we did learn when we aggressively protected aphids is when we made several aphid sprays, it was almost a given we were going to flare spider mites. So we had to overspray most of our trials with a miticide as a result of disrupting beneficials with these aphid sprays. So in summary, at least in the trials we've conducted the last 2 years, aphid management did not influence the incidence of cotton leaf roll dwarf virus in these trials. And again, we got a lot of comparisons where we sprayed very aggressively each week compared to an untreated. And again, they tested positive for the virus. We saw very little symptomatic plants or very little disease from the virus in these trials. Aphid management did not increase yield compared with the untreated. And finally, aggressive aphid management flared spider mites in Georgia and Alabama. And with that, we'll close.